Hello, everyone, and thank you for joining us for today's webinar on best practices in somatic CNV detection. I am your moderator, Brenton Pyle, and we are very excited to have Eric Talovich reviewing the best practice methodology behind CNVkit. If you have any questions during the webinar, you can submit them through the chat or the Q&A sections at the bottom of your Zoom window. We'll answer as many questions as we can in the questions portion at the end of the webinar. All right, now without further ado, I'll hand it over to Eric to kick it off.
Thanks, Brenton, and thanks everyone for tuning in, and let's get this started. So when I say best practices, first of all, that means not necessarily the only practices. There are other things that you can do for copy number detection that won't necessarily be covered in this presentation, but we will be happy to answer your questions as we go along. So just keep those questions coming, and hopefully we'll get to them. All right, first of all, we start with the basic problem, NGS screening for cancer. We're looking for copy number alterations of a variety of different kinds to use for a variety of different signals for tumor diagnostics, prognostics, and copy number analysis can help with all of that. Another constraint of NGS screening is that for accurate detection, we tend to require deep sequencing. I'll go into the details of that in a minute, but the constraints we're working with typically in the clinic are low input DNA, low sample purity, even going as far as circulating tumor DNA, just 1 or 2% tumor DNA in the entire sample. And we're also interested in hopefully detecting subclonal alterations in the tumor tissue. Even within the tumor cell population, driver mutations are not necessarily present at 100% of the cells. So all of this means we need to make the most of the DNA that we have. That's why we use targeted sequencing, either exome capture or a selected gene panel of cancer-related genes. That's the standard of care that's applied at major medical centers like Memorial Sloan Kettering, Dana-Farber, UCSF, and companies like Foundation Medicine. Targeted sequencing brings along a couple of pros and cons. Specifically, we get deep coverage over a fraction of the genome. That makes SNV, indel detection easier and helps us detect tertiary characteristics like microsatellite instability. However, structural variant and copy number variant detection is a lot harder, mainly because the breakpoints surrounding these alterations are rarely captured in the target panel. If you look at the table at the bottom of this slide, I show the characteristics of each of the major sequencing technologies. With whole genome sequencing, we cover a sequencing footprint of essentially the whole sequencing accessible genome, 2 to 3 gigabase pairs, depending on your level of ambition, and filtering for problematic regions. In practice, that typically gives you a depth of coverage of 30-fold, which is fine for population sequencing. On the other hand, if we want to go a little deeper, we can go with whole-exome capture. That's a sequencing footprint of 30 to 60 megabase pairs, just 1 or 2% of the genome, but we get a much deeper coverage depth, 150 to 200-fold coverage, which, as I said, helps with SNV detection and generally detecting actionable variants. We can go a bit further with that idea by designing a target panel for just the genes that are associated with your disease of interest. There are about 500 genes that are associated with cancer and actionable or informative in some way. So a target panel of those might give you a sequencing footprint of 2.3 megabase pairs. That's 0.1% of the genome, 1,000th of the genome, but you get much higher depth of coverage, 500-fold. Given the normal variation in coverage across the genome, that can still give you the power to detect subclonal alterations in impure tumor samples And that's the kind of sensitivity that we want for clinical applications. So how do we resolve this problem with structural variant and copy number variant calling? For structural variants, since we have a clinical application in mind, there may be known fusion breakpoints that we want to be able to detect, and we can just live with the fact that we're not going to detect every structural variant in the genome. So we include in our capture the introns of those fusion breakpoints within the oncogenes of interest. We can confirm those alterations with RNA-seq or FISH later if there's an ambiguous result or a suspected alteration within a gene. For copy number, that still doesn't get us quite there. We want, we'll accept coarse grain coverage, but we want it across the entire genome. So one solution is to scatter small targets over intergenic SNP sites. This is fairly common practice. These SNP sites are not necessarily useful for diagnostics on their own, but in aggregate, they can give you coarse-grained copy number, maybe at the arm level, chromosome arm level. And by targeting SNP sites, we can probably get a signal for loss of heterozygosity if it's present. Doing all of this is estimating copy number from the on-target read depths. These are the captured regions, either the exons of targeted genes or these scattered small targets. And if we have an ambiguous result, we can confirm it with array CGH or FISH. It's still not entirely satisfactory, though. So we're looking for a better way. To start, we'll go to the source, a method that does work and is in common use in the clinic. That's array comparative genomic hybridization. The way it works is a microarray method. We measure microarray probe hybridization intensities at points across the genome, a few 100,000 points. From those probe intensities, we compute the ratio of intensities versus a baseline. That gives us, we can put these to logarithmic scale for reasons we may or may not go into later, and those are your log ratio values. That's what you see in the plot on the right as gray dots. Log ratio probe intensities across the entire genome. The x-axis here in the plot is chromosomal position, and the y-axis is the log ratio of probe intensities. Now we can join these similar looking regions together through a method called segmentation. That gives us discrete copy number change points. So in chromosome 1, for example, we see that the short arm, the P arm, has maybe a slight copy number loss, or it could be copy number neutral, whereas the long arm, the Q arm, has a copy number gain. The log ratios cluster up about 0.58 maybe, and that would imply a single copy gain of that arm of chromosome 1. And we repeat that logic across the genome, connecting these probes with similar log ratio values. And that gives us a copy number profile. Within each joined contiguous segment, we calculate a mean copy ratio, a mean log ratio, and that's something that we can report. So how do we accomplish the same with next generation sequencing? Our starting point is mapped reads to a reference genome. And from those mapped reads, we can easily calculate a coverage profile. Here I've shown you an example of a targeted gene. This is BRAF, the gene BRAF. The bottom track here is the gene model. These rectangles are exons that have been targeted. You can see the coverage up here on the higher track piles up around each exon. It's obviously much higher at each exon, and then there's a little bit of spillover on each side. Then we get to a point where we've captured the introns of some of the introns of BRAF as well. But the depth of coverage here is about half of what it was at those introns, at these exons on the left, which implies there's a copy number breakpoint here, and the copy number in the genome for this right half is about half of what it was for the left half. Maybe there was a loss of one half or a duplication of the other half. So that's one way to do it. But there's another feature here that's interesting, and I want to point it out. It's that even in these untargeted regions, not the captured introns, but the uncaptured introns, we see some reads aligning here. Now, it might be initially unclear whether those are just random noise in the data. Are they misaligned reads? Or are they DNA that kind of slipped through during target capture, during hybridization in the lab process? Actually, it's the latter. What you have is effectively very low pass whole genome sequencing on top of your targeted sequencing, and you do hybrid capture. And we can use that information. Here's an example of a gene zoomed out a bit, where you can see both processes happening. This is a gene with a very high level amplification. We can see at the exons that were targeted, here, here, and here, we see sharp spikes in coverage. The coverage axis here is in log scale, so if you see a point popping up above the rest, that means it's much, much higher than the baseline. So here we're going up to maybe 6,000-fold coverage on target. But in the adjacent off-target regions, the introns and a little bit of the intergenic area on either side, we see that these off-target reads are not at this random scattering of maybe 1-fold coverage. It pops up to maybe 5-fold coverage or higher. So let's use that information. It looks like those are both valid signals. On-target coverage and off-target coverage are valid signals of copy number. So here's the basic method. We look at the middle box here. You've already seen the coverage track and the gene model track. Here we've tried to replicate array CGH. The little green dots are on-target coverages converted to log scale. We've already normalized this to a baseline. So well outside the amplified region, you'll see that the green dot is close to 0, which is neutral in log scale. Then these green lines represent the off-target average read depths within each interval, what we're calling bins. And again, we see that well outside the amplified region, the off-target binned log ratios are close to 0, close to neutral, and they track pretty closely to the on-target bin log ratios. Within the amplified region, within the vicinity of MITF, we see that on-target log ratios jump way up to about a level of +4. That's a copy ratio of 2 to the 4th, or 16 at least. So this is a 16-fold amplification supported by both on-target coverages and the adjacent off-target coverages. They track pretty closely, and this is without performing any additional normalization. So this is a very strong signal. We see that the coverage right at the edge of this falls off a bit. That's because it's averaging both the amplified region and the neutral region. And that's just a signal that we're working with more coarse-grained information. And so we have to take that into account. All right, let's put all of that into action. We've implemented this in CNVkit, which is a standalone software package and also an app on the DNAnexus platform. Both work the same way. If you follow the legend here, the input files are in pink, Most of these are actually optional, and the app will fill these in for you as you go if they're not provided. But here's the workflow for targeted sequencing and exome sequencing with hybrid capture. You start with your mapped sequencing reads. Those are your BAM files, one per sample. So for a given tumor sample, we have our BAM file, and we have the bed file from the capture kit. These are the targeted regions, actually the captured regions, if you can get that file from your vendor. You provide that bed file, hopefully annotated with gene labels, and your tumor sample into the pipeline. If you've done off-target binning yourself, you can provide that here. Otherwise, the app will calculate it for you. And then you have your coverages within each bin, either on-target bins, which are basically just exons, sometimes subdivided for consistency for large exons or targeted introns, and then your off-target bins, the uncaptured regions, which have already been subdivided into roughly equal-sized regions. So there you have coverage depths within each bin. We apply a few normalization steps, which I'll get to possibly a little bit later. Then do further normalization to a reference copy number profile, which I'll discuss in just a second. Perform some automated bias correction, and there you have your copy ratios, which you can treat more or less just like array CGH, just as it would come out of the lab. Which means we can apply the same segmentation algorithms to get discrete copy number segments, and that's your genome-wide copy number profile from targeted sequencing. And there's a lot you can do with that. We can visualize it, perform some downstream analysis, and export to other formats so that it's compatible with other software. All right, now I think we're ready to get to the parts that I skipped over. There's an initial configuration step that you can go into as much or as little as you'd like to when running the CNVkit app or the software itself. Starting with the target bins again, the capture region bed file from your vendor. We look at the reference genome. Having looked at the regions that aren't masked out by N characters, this is usually the centromeric regions and maybe the telomeres. We divide those regions that weren't captured into bins. That's how we generate off-target bins. We can reuse that file as input later. We can also keep going by using those target bins and off-target bins with the BAM files from matched normals or even unmatched normals that were processed with the same lab protocol. So if you use the same capture kit to process tumor samples as you did to process matched blood normal samples, then we can use those normals to build up a reference profile. Now, it actually works better not to use a single matched normal for each tumor sample, as you would with Somatic CNV calling, but rather to pool your normal samples together. And CNVkit will do that automatically for you. You just provide all of your normal samples in one group, and it will calculate the bin coverages, run through half the pipeline, and consolidate all of this information into a reference profile. That reference profile has the average normal coverage at each of these bins across the entire genome. In addition, if you have multiple normal samples, it will take the variation, sort of the variance or standard deviation of each bin's coverage across all of the normal samples. So the first bin of chromosome 1, what is the distribution of coverages in all of the normal samples? Are they all pretty close together, which means it's a very consistent copy number signal, or are they kind of scattered all over the place? First bin is probably close to a telomere. It's probably all over the place, so we'll assign a high spread value to that bin and assign it a lower weight because it's a less reliable bin. For the more reliable bins across the genome, where the normal samples agree more closely on coverage, those will be upweighted. And we'll use that information in segmentation too. There you have it, a reference profile that's reusable and captures all of the information from the target and off-target binning, as well as the GC content across the genome, sequence repeats, and we can use all of that information each time we process a new tumor sample to, again, normalize the bin coverages, perform automatic bias correction, and derive a new copy ratio file for each tumor sample as it comes in. So let's try it out. I'll show you a nearly live demo on a set of samples that were provided to the public by Baylor College of Medicine. These are 7 tumor normal pairs that were sequenced and prepared by exome capture to 100-fold on-target coverage. The off-target coverage is about 1-fold, but you don't really need to know that to run this analysis. So we're going to use those 7 normal samples to build a pooled reference. Then we'll analyze each of the 7 tumor samples in parallel. We open up our DNAnexus project, the TCRB samples. To save time, I've already run this job, so I can quote you a walk time. And we'll just click through it and see how the whole job ran. Here we see that we used not just one machine, but actually a bunch of independent workers on the cloud. This one was run on the Amazon cloud. So we used initially a single entry point, then applied a scatter-gather approach to run the coverages of each normal sample in parallel. Then we collected these into a reference profile in a separate step that ran for about 10 minutes. Now remember that this initial configuration, this building the reference, only needs to be done once per lab protocol. So this half hour spent in collecting the normal samples, analyzing the reference genome, and building a reference, you can subtract that from subsequent runs for any additional tumor samples you want to process with the same reference profile. And that's actually the recommended practice. So how do we process those individual tumor samples with the reference? At about 20 to 25 minutes a pop, we run each of those tumor samples in parallel, again using a scatter-gather approach. Finally, do a little bit of post-processing to make this output nice. Consolidate all of the plots into multi-page PDFs that you can download more easily. And we return a variety of results to the user. Again, to recap, the inputs were just the 7 tumor samples, the 7 normal samples, the capture targets, the reference genome sequence in FASTA format. And since these are tumor samples, I opted to drop very low coverage bins. That's using the assumption that since these are not pure tumor samples, even if there's a deep deletion, a total loss of a gene or a genomic region in a tumor sample, in the tumor cells in a tumor sample, that sample will still have some normal cells that do contain the DNA at that gene position. So the coverage at that gene will not be 0. It will be a little bit above 0, and all you're capturing are the normal reads. We can deal with that normal cell contamination pretty easily in the pipeline. I'll show you how in a minute. But the principle is that when you do see a bin with 0 coverage, it's not real. It's just a part of the genome that wasn't captured effectively or wasn't sequenced effectively, or maybe was too repetitive to map reads to. So we drop those bins. We know they're garbage, we just throw them out. So the outputs. The segmented files are tabular files. The extension is .cns, copy number segments. That's the easiest thing to work with for analysis, although it drops the bin level coverages. The app generates a few helpful plots for you, a heat map of the entire cohort, some QC metrics, the copy number reference built from those normal samples so that you can reuse it for later analyses. Some more summary stats, genes with breakpoints in them. We've exported to seg format, which is a more standard format that you can load in IGV or UCSC genome browser. Some unfiltered segmented calls, gene level calls, those original copy ratio values that I mentioned, and the multi-page PDF of plots for each sample. Oh, we have time. Okay. We have, I think, enough time to quickly look at the log files to see exactly what the app did and recap that workflow that I showed you, the simple diagram. So the first step is to scan each chromosome for the sequencing accessible regions. This means we track which letters are N, basically, in the genome sequence FASTA file. Those are the masked out regions that were not part of the HG19 assembly or not considered sequencing accessible. We do that for each chromosome, including the alternative contigs. Now, for just a couple N characters in a row, we probably don't care too much about masking that out because we're using coarse grain coverage. So we'll iron those out a little bit. Now we're ready for the next step. We choose one mid-size normal sample if you provided normal samples and use that to quickly estimate the on-target coverage and off-target coverage. We'll use that information to calculate a reasonable bin size if you didn't specify it. So here we have a summary table. The on-target read depth is about 100-fold. The off-target read depth, what we're calling anti-target, that's about one-fold coverage across the whole genome. And we use those numbers to calculate bin sizes. We use a maximum bin size of just shy of 1,000 for targets. That won't involve chopping up most exons, but for tiled introns, we'll use that threshold to break them up. For off-target bins, that's 90 kilobases. And that tells you your resolution in the output of this. From that point, we have everything we need to fire off some subjobs to calculate binned coverages in the normal samples and build a reference. So coverage calculation is really the only computationally intensive step of the pipeline. That's why we break it off into a separate job and do it in parallel. We do each sample in parallel, and additionally, the coverage calculation is also parallelized. Here we use 8 CPU cores within each cloud worker. And we use the optimized SAMTools implementation to calculate coverage depth. So that's how you get coverage across the entire genome in just 13 minutes. Finally, we aggregate all of those into this reference job. We've already calculated our binned coverages for each normal sample. Now we do some sanity checks. One heuristic, which is pretty important, is to check the genetic sex of each sample. Are there 2 X chromosomes and 0 Y chromosomes, or 1 X and 1 Y? We want to be able to call copy number on the X chromosome. So we need to sort out a mix of control samples that may be both male and female. By default, we'll make X look like it's always diploid. So for samples that look like they might be male, one copy of X, one copy of Y, we'll double the coverage on X before averaging them together with the female samples. This can also be specified on the command line internally, but the heuristic here is pretty effective. So we go with the route of just letting CNVkit guess and then log what the guesses were. So if you see anything weird in your output, you can track down what happened. Now we extract GC content and sequence repetitiveness from the reference genome for our automated bias corrections. Full details of that in the paper. And then we do our QC for each bin. As I mentioned, we average together the correct coverages for each bin across all of the normal samples. That gives us our average log2 ratio at each bin, and then also a robust estimate of the standard deviation of those coverages within each bin. If the spread is too high, over 1, then we consider that bin just much too noisy. If the coverage depth is below -5 on log2 scale, so less than 1/32 of the genome-wide average, then that's a very poorly captured bin, and we'll reject that too. These are fairly loose criteria, so we're only rejecting less than 2% of the targets in the sample. And for exome capture, that's pretty good. Exome capture is basically unbiased, so you're attempting to capture genes that are maybe a single exon in weird genomic locations. We expect some noise to come out of exome sequencing versus a curated target panel where you already expect the genes to be well behaved. So here's a comprehensive report of the rejected bins, target bins, in case you're wondering why a particular gene is missing data. Generally, you'll just be missing the exon of it, a particularly bad behaved exon of a gene rather than the entire gene, but for comprehensive support and troubleshooting, here it is. Here's all of it. For off-target bins, we don't do the same because you weren't interested enough to capture those bins in the first place. You're probably not too concerned about which off-target bins in particular were rejected. And here we see that more of those bins were rejected under the same criteria, either too low coverage or too much variability in coverage across the normal samples. Still, that's only about less than 8% of the off-target genome that was rejected by this default QC. So the rest of the off-target genome is still available for copy number analysis. And that's how you build a copy number reference. That reference is reusable for every tumor sample, so we just dive right in. For each tumor sample, we apply all of the information in that reference profile. The average expected coverage, the GC content of the bin, the sequence repetitiveness of the bin, and the spread of the bin is already converted to a weight value, so we'll copy over the weight value for that bin. Here we see that after processing, the off-target bins are twice as variable in coverage as the target bins, so we'll downweight the off-target bins in this sample accordingly. We're just adjusting the weights again. So that's about 350,000 bins, which we then apply segmentation to get discrete copy number regions. We'll use the standard method CBS circular binary segmentation, the same thing that's normally applied to array CGH. In parallel, we do a little bit of signal smoothing, apply segmentation to get these piecewise constant regions, and write out our CNS file, copy number segments. Here, out of 350,000 bins, we extracted 80 segments. That cleans things up a lot. And we immediately go ahead and use both the bins and the segments to draw a scatter plot. I'll show you an example of that in a minute. We write a diagram. This is basically an ideogram or karyotype if you want. Just a couple of visualizations to help you understand the data. For each segment, we calculate some additional metrics using the copy ratios together with the segments. The key one here is confidence intervals. We use a loose interval. We sample the copy ratios by bootstrap to estimate lower and upper bounds for the segment mean. And then in the next step, we use that segment mean here with the filter CI. And if the lower or upper bound for a particular segment crosses 0, meaning the segment could plausibly have just been neutral, then we basically erase the segment breakpoints and merge it with the adjacent segments so that it basically becomes neutral in the output. So what's left after this is a neutral baseline plus copy number alterations that were supported by confidence interval analysis. We also recenter it so that the neutral values, instead of being close to 0, are directly at 0. If this goes wrong without this step, you'll know, but I won't get into it. And the output of all of that is your call.cns file called copy number segments. Now, we don't know the tumor purity here, so we're using another heuristic method, threshold, for assigning integer copy number values. This is a cutoff of if it's above 0.2, well, if it's below negative 1.1, it has a copy number of 0, deep deletion. If it's between that and minus 0.25, has a copy number of 1, meaning single copy loss for diploid chromosomes. If it's between that and positive 0.2, then it's neutral, 2 copies. If it's above 0.2, 3 copies, and above 0.7, we'll call it 4 copies. It's just a dense form of representing all of that information. So that's our thresholding method. If you had instead provided the purity of each tumor sample going into this analysis, it would instead do a little bit of algebra to determine what the threshold should be for, given that tumor cell purity, what would integer copy numbers look like in log2 scale. Basically just applying 2 to the log value and rounding up or down to the nearest integer. Get your gene level reports and report the genes that have breakpoints in the middle of them. And those are all of our output files from the single tumor sample analysis. Those plots, the scatter plot and the chromosomal diagram, get concatenated for all tumor samples that go into the analysis. I won't show you that job log. We also aggregate the outputs, some of the other outputs, to generate a seg file of all of the CNS files, all of the segments, all into one file that you can load in a genome browser. That's a standard format. We also plot the segmented values across all samples as a heat map. And for your triaging, we calculate the genetic sex of each sample and give you a report of that as well. And that was it. Well, that took 1 hour and 10 minutes to run and slightly less than that to walk through the log. So what else do we do with these outputs? There's a lot of downstream analysis you can do with it, actually, besides just trusting the gene level reports. You can run CNVkit interactively on the DNAnexus platform in our cloud workstation app or in a Jupyter notebook if you prefer. When you're in a Jupyter notebook, you can also use the built-in plotting mechanisms. I'll show you some of the plots that you can generate either from the app itself or through this interactive mode. Now I'll explain how each of those plots can be used to understand the genomes you're looking at. So here's an example heat map that comes right out of the app. This is not the TCRB samples from Baylor. This is a different cohort that was analyzed in the published paper on CNVkit. TR is targeted resequencing. We have, I think, 42 samples here. So the x-axis here is genomic position. The y-axis is samples. And you can see some of the samples are a bit noisier than the others. Some of them have widespread copy number alterations. Sample 55 has whole chromosome losses across much of the genome. And you can see some recurrent alterations too. I won't go into too much about what those mean, but I'll focus on one specific alteration that's recurrent in 3 samples. This is on chromosome 12. If you zoom in a bit, you'll notice that around positions 60 megabases and maybe 70 megabases on chromosome 12, there is sometimes a large-scale copy number loss, not a deep deletion, maybe single copy. But on top of that, you see these deep red regions. That means amplification, not just single copy gain, but something higher than that. It's most obvious in sample TR95, but we also see the same pattern in 37, and that second gene around 70 megabases shows up in sample 6 as well. So what are these recurrent alterations? We're going to use another plot type, the scatter plot, to understand that better. But first we're going to look at the diagram. This is the chromosomal diagram or ideogram that I mentioned. Just another way of viewing the heat map, this time with gene labels. Sample 95 that I mentioned as the most interesting case. We're looking at chromosome 12. We see that single copy loss with gene amplifications at 3 points. We can follow the gene labels to those points on the plot, and I see CDK4 and MDM2. I know those are oncogenes, so I'm going to take a closer look at those. Here's the scatter plot that we're going to use to analyze those. It's another view of the data. This is the whole genome view that comes right out of the app. Fully zoomed out, you can see some large-scale alterations, gain of chromosome 1p, loss of chromosome 10, and then something complex happening on chromosome 12. So finally, here we are. That region of interest on chromosome 12 in one sample. With the scatter plot in interactive mode, if you specify the genomic region of interest, it will zoom in on that chromosome and label the genes in your selection. So here's CDK4 and MDM2. The baseline looks like a copy number loss. It's below 0, but not a deep deletion. And then we see a lot of supporting data points and segmentation around these 2 genes, CDK4 and MDM2. For cancer samples in particular, for bulk tissue, solid tumor sequencing samples, this is what you're seeing here with 3 segments. This kind of pyramid shape is real. what happens is that a somewhat larger genomic region is gained earlier in the tumor's life cycle, basically. That gives it a selective advantage. Then subsequent genomic alterations cause duplications and amplification, more specifically on the gene that's serving as a driver, giving those even further selective advantage. So the copy number gain kind of piles up and focuses in on the gene that's really driving tumor growth. We see that here with MDM2 and CDK4 happening the same way. What else can we do? If we provide a VCF file as app input, we'll also get a plot of the SNP allele frequencies at the same positions across the genome. Here's an example, not the same one from the same cohort, but a different one where we see some large-scale alterations that I will just quickly point out. Chromosome 1 is lost, chromosome 2, also a single copy loss, a little bit of chromosome 3, and I see losses on 10 and 13 as well. If I go down to the variant allele frequency track, I see allele frequency is shifted in the tumor sample from the expected 0.5 up to about 0.7 on chromosome 1 and 2, a little bit of 3, 10, and 13. Sure, all of that's consistent. When one of the alleles is lost in the tumor sample, you'll see a mix of basically heterozygous normal cells and homozygous tumor cells, and so there's allelic imbalance. But we also see allelic imbalance on chromosome 6 and 9. What's going on there? So in those tumor cells, we see neutral copy number, 2 copies of the chromosome, probably. That means that there's still allelic imbalance. There's still a copy number shift, so loss of heterozygosity. And the reason that isn't fully 1.0 is because of the normal cells in the sample. So the tumor cell population lost 1 copy, leading to loss of heterozygosity, and then gained a copy again. So from total copy number, this is imperceptible, but once you add in variant allele frequencies, you can see that there were alterations on chromosome 6 and 9, which could be clinically meaningful or actionable. Finally, I mentioned that you can use the tumor purity input to the app to automatically adjust segments. Here's what that looks like in practice. The gray dots are before the tumor heterogeneity correction. We see that this tumor sample was maybe 50% tumor cells, 50% normal. That's typical. Then we applied this purity correction in the call command at the end of the pipeline. This is just built in when you use the app. Just provide the tumor purity estimate as an input, and we'll do this automatically. The segment means are shifted according to tumor purity, and we get a value of, instead of this kind of ambiguous value of around -0.5, we have -1, which for a diploid genome translates perfectly to single copy loss. That's easy to interpret. Single copy loss of chromosome 1, 2, part of 3, 10, and 13. And I'll quickly go over, I'll point you to some validation of this work to give you more confidence in using CNVkit for your own analyses. We've compared it to array-CGH directly, just a head-to-head comparison and analysis by next-generation sequencing and CNVkit versus array-CGH using the standard lab methods. And we can see here on this cell line with severe aneuploidy, the copy number profiles visually match almost exactly. If we zoom in a bit, head-to-head on genes of interest, ALK and RET, even at the gene level, NGS and CNVkit closely match array-CGH. If we do a third method, FISH, see that the absolute copy number for this hexaploid genome matches closely with array-CGH and CNVkit. The same is true for a copy number loss at RET. Can we catch focal deletions, and what does that look like? The targeting at CNVkit is a little bit more sparse, but we still pick up a deleted gene very clearly. And the breakpoints for that focal deletion match closely with array-CGH, even though array-CGH has much more even binning and therefore denser binning in the off-target regions. Binning, I mean probes here for array-CGH. The full details of all of this are published in a paper in PLOS Computational Biology. This is an open access article, so you can just go right to it. I gave the DOI a little earlier in the presentation. You can also just search for the title. That's where we fully validate this on exome and targeted gene panel samples. Another more recent publication did the same work for targeted amplicon sequencing. This is a PCR-based method where there are no off-target reads to supplement the targeted read depths, but the research team found that it was also still acceptable for clinical use.
All right, thank you, Eric. Before we get started on questions, I did want to mention in the follow-up email, we'll be including links to those papers that are publicly available, as well as the workflow. I know a couple people asked about that, so I just wanted to address that. All right, so our first question is on bin size. Question asks, bins are irregularly sized. Can you comment on how the bin size is set?
Sure. So the average bin size is chosen separately for on and off target bins. That's done by the auto bin command in the app. But the way that works under the hood is it quickly samples the genome for, or a representative normal sample for on target and on off target coverage depth. And using that information, it aims for an equal number of, it aims to choose a bin size so that an equal number of bases or reads is captured within each bin for both on and off target coverage. So for 100-fold versus 1-fold on and off target coverage, like we saw in the Baylor samples, you would expect the off target bin size to be 100 times the on target bin size. Now, since there are some regions of the genome that are masked out, we subdivide all of the contiguous regions into what is as close as possible to equal sized bins. So we choose the number of bins to squeeze into a genomic span so that the average bin size of those subdivided bins is roughly the same. Then, since those might not be exactly equal, we just assign weights. We just change the weight value for the bins up or down to compensate for whether those bins are a little bit too big or a little bit too small.
All right, next question is, doesn't this method assume uniform coverage across all bins? Often probe tilting is not uniform. How does CNVkit handle non-uniform probe tilting? Or tiling, sorry.
Uniform coverage across all bins. So I don't know if you mean, did we do a two-track approach to capture where some regions are strongly enriched, some are just enriched half as much with half as many tiles per kilobase? If it's the latter, then you're relying almost entirely on building a useful pool of normal samples and a useful reference profile. And if you have done that, then the reference profile will capture, will encapsulate the information about which bins are expected to have high coverage versus low coverage.
All right, next question is, do you have any additional recommendations for amplicon/gene panel when you have multiple amplicons per target?
Multiple amplicons per target. See, there is another program out there called CNV that I've seen in widespread use. From this other validation paper that I pointed to at the end, they seem to have good results with using CNVkit on amplicon data, even without specifically accounting for the locations of the amplicons.
Okay, next question is, does the aligner used affect the coverage calculation output?
Does the aligner used? Most likely, yes, but well, in a sense, yes, but that's kind of the sense that you would expect. Wherever the reads go in the BAM file, that's where coverage will be calculated. So CNVkit just uses the SAMtools pileup engine. It uses PySam, which wraps HTS-lib, so this doesn't create a separate file, but it does use the pileup engine. So wherever you see the reads aligned in your genome browser or IGV, that's where coverage will be calculated. Your different aligners may choose to put the reads in different places, but the calculation method is the same. If you have a tumor normal sample swap, first off, that is a risk, and you should have several safety checks in your pipeline to account for that. If you've used a pre-built pooled reference, then you'll immediately see that the copy number profile of the normal sample has alterations that aren't in the tumor, and that will set you off. You can do the same check with SNVs. You see somatic SNVs in the normal sample that weren't in the tumor sample. Those should be part of your pipeline QC. Now, what if you just did a paired run with the CNVkit app, one tumor sample, one normal sample? You would basically have inverted CNV calls in the tumor sample, which is really the normal sample. And if you did it that way, without your pre-built reference, you might see a lot of bins filtered out for having low coverage or overly high coverage. The reference profile step will catch some problems, but your results will be hard to interpret. So I strongly recommend building the reference. The reference profile does a lot of work in sorting out inconsistencies in your pipeline. That's kind of a follow-up to the other question just before this too. If there are inconsistencies in the way that you tiled your amplicons or captured your amplicons, if you use a pooled reference, it will all get ironed out in the end.
Okay. Our next question is, can we use CNVkit to detect exon level copy gain loss? This
isn't built into the app currently. There's a script in the upstream project for calling exons. It's called CNV test. And integration is an ongoing project. So I hope to see that in the DNAnexus app within another 1 or 2 releases, so a few months out. There's also a CNV app on the platform called CLAMS, C-L-A-M-M-S. This is specifically for exome sequencing. It doesn't use off-target reads, but it does have a single exon test built in. So you might try that if CNVkit isn't working out for you and you need your answers right away.
All right, moving on to the next question. Do you think modeling the purity ploidy estimation together with the ASCN calling is a better strategy?
Titan CNA does that pretty well, or Titan CNV does it all in one shot. And you can use the output of CNVkit as input to another project called Pure CN, which does the same thing. So general answer, yes, kind of, if you really need that. I'm assuming ASCN is allele-specific copy number. for those on the line. So yes, you can think of CNVkit as kind of a toolkit for building up to that, if that's what you need. In clinical use, typically they just look at the log ratios because that's what they're used to doing with array-CGH. And sometimes you don't really need to know the exact integer copy number of all alteration. You just need to know that this known oncogene is amplified or this known tumor suppressor is lost.
Okay, moving on to our next question. How does CNVkit handle using a normal cohort created using samples sequenced at a lower depth and tumors sequenced at a higher depth when normals and tumors use the same capture kit?
It handles it just fine. That shouldn't be a problem unless you're doing truly low-pass sequencing for your normal samples, or basically a lab protocol that looks vastly different. The coverage profiles for each sample are normalized to the genome-wide average before doing anything else. So if one of your samples has 100-fold average coverage and the other has 200-fold average coverage, they'll both basically be rescaled to the same baseline so that they're comparable. And all you're looking for after that is relative changes from the baseline.
All right, our next question is looking for some recommendations. We're wondering if you recommend what you'd recommend to use to annotate CNVs, and if you'd recommend using other CNV callers to have a more accurate batch results.
So there's some kind of bed tools-like functionality built into CNVkit. You can read the documentation to see exactly what options are available. There's a lot of flexibility, more than I really want to talk about, but yeah, it's actually pretty reasonable and common to combine CNVkit results with not only other CNV callers, but also structural variant callers. That's probably the best practice overall, to run CNVkit alongside another SV caller like Manta or Lumpy. I want to put in a plug for the BC Bio next-gen pipeline, which makes some of this easier. We offer that on our platform as well, but if you want to see some best practice pipelines laid out for you, that's a good starting point.
Okay, our next question, for your test, what was the CNV threshold? How many copies? Also, how large is the copy in BP?
Oh, how large is the copy? Let's see, for the Baylor samples that I showed you, the CNV threshold was the built-in one, which is pretty permissive. It assumes a tumor sample purity of at least, I think, 30%. I think this is in the documentation online, but so above plus 0.2 log ratio, we'll assume that's 3 copies or more, and below minus 0.25, that's 1 copy, a single copy loss, or further below that, a deep deletion. How large is the copy? In the .cns file, the first 3 columns look like a bed file, so that gives you exact breakpoints, and you can just do subtraction, the endpoint from the start point from the endpoint to get the region size. But note that because of this binning strategy, those aren't exact breakpoints. Those are imprecise breakpoints in VCF terminology. You can also export the .cns files or the .call.cns files to VCF format directly, and that might be easier for you to interpret. But yes, it does give you the exact coordinates and implicitly the size of each segment that's altered. Why don't we run a matched normal? You can see the gory details of this in the paper, but basically the strategy of using a matched normal for copy number calling is kind of grandfathered in from somatic CNV calling, I think. Whereas in CNV calling, you're starting with a genome that's chock full of SNPs. So you're kind of lost in a wilderness of alterations from the reference genome. And your first step is to subtract out all of those before focusing in on this much smaller number of somatic SNVs. For copy number, you're starting with a reference genome that's pretty close to flat. I'm discounting the smaller scale population level copy number variants, which is probably the more common definition of what CNV is anyway. What we're really looking for here is copy number alterations, CNAs, at a somewhat larger scale. So instead of subtracting out the CNV equivalent of SNPs, you're subtracting out just normal coverage variation introduced by your lab process. And you can model that lab process a lot better by pooling together multiple normal samples rather than just using a single one. With just a single one, you're basically subtracting a lot of noise from a lot of noise, and the result will still be noisy. And yes, if you can get FFPE normals, that is ideal. If you use fresh frozen normal samples to build your control pool, that is still usable for analyzing FFPE tumor samples. It's not ideal, but it's usable.
All right, we're about 10 past the hour here, so I think that'll do us for today.